Hello and welcome to MCDB 1161, Phage Genomics Lab 1. My name is Christy Philman and I'm one of the instructors for this course. In this video I'll go over the syllabus for the course, then give you a tour of our Canvas site and show you where to find important information on Canvas, go over some lab etiquette, and finally discuss how to write a lab notebook for this course. A PDF version of the course syllabus that looks like this version can be found on Canvas by clicking on the Syllabus PDF link on the home page or going to the Course Information folder in the Files section. Each week is divided into a Lab A period the first time that your lab meets for the week, either Tuesday or Wednesday, and a Lab B the second time that your lab meets for the week, either Thursday or Friday. There's also a Wednesday lecture that is attended by all Phage students at 3 p.m. The reading assignments for each week is listed in the first box of the syllabus. Links to videos and articles can be found on Canvas. The first column of the syllabus notes the lab period or date of the Wednesday lecture. The second column notes the topics covered in class, and the final column notes the assignments that are due that day. Most assignments are due at the end of your lab class on the day noted on the syllabus. Pre-class assignments are due right before lecture, 3 p.m. on Wednesdays, and writing assignments and problem sets are due on Fridays at 11:59. Your final grade will be calculated out of 537 points. The delineation of these points are shown on this chart, and I will briefly describe each category of points. Clicker points and lecture participation. On Wednesdays during lectures, we'll ask several clicker questions, and you will earn clicker points for using an iClicker in class. If you earn 80% of the clicker points based on your participation, not on correctness, you will receive all 15 possible points. 2 absences are allowed, and your answers must be recorded electronically to receive points. Starting the second week of class, we will form lecture groups of 3 to 4 students. In your group, you'll be asked to discuss topics, answer questions, or complete activities which are submitted to Canvas. Groups will also be called on to answer questions during lecture. These activities are worth 10 lecture participation points. Pre-class assignments cover assigned reading material for each week and are due right before lecture on Wednesdays at 3 p.m. There are 12 pre-class assignments, each worth 3 points, and your best 10 scores count towards your grade. Problem sets are based on reading, lecture material, and lab activities, and are due weekly on Fridays at 11:59. See the syllabus for dates. Each problem set is worth 7 points, and your best 10 problem sets count towards your grade. Combine the pre-class assignments and problem sets are worth 100 points. Lab notebooks are worth 5 points weekly for a total of 50 points and are due at the end of the lab class on period B. Lab notebooks should mostly be completed in class as you are working on your experiments. I will discuss the lab notebooks in more detail at the end of this video. Lab activities are worth 120 points. Most lab activities will be completed in class and turned in at the end of that lab period. You are welcome and encouraged to work on lab activities as a group, but be sure each member participates in coming up with the answer and that each group member writes their answer in their own words. One of the projects in this class is writing a scientific paper. We will discuss in detail the format for this paper and give you opportunities to turn in drafts of the paper for feedback before you turn in your final paper. Together, the drafts and final paper are worth 62 points. The Cure Symposium is a great opportunity that Phage students have to present their research to a larger audience and to hear about other research projects conducted by student researchers at the University of Colorado. Attendance at the Cure Symposium is required, and you will receive 60 points for preparing a scientific poster and participating at the symposium. Finally, there will be a midterm and final exam. The final exam will be on the scheduled final exam date for the class. These assessments will cover material from the readings, lectures, and lab activities. The labs will be open at scheduled times for you to come in and work on your experiments. This is a great opportunity to repeat experiments and continue with your experiments so that you have time to work on an independent experiment later on in the semester. LAs will be available at all open labs to answer questions and to help you work on your experiments. If you're interested in working on your research outside of class and potentially designing your own experiment, Open Lab is a great opportunity available to you. Talk to potential lab partners on the first day of class to see if other students share your interests and would like to partner with you in the lab. Open Lab is also available for students to get help with writing, problem sets, or other assignments, and to make up absences. A large portion of this course requires your attendance. Every student is allowed 4 absences for any reason. These absences do not need to be documented. Absences must be made up by attending an open lab time. Absences that are not made up within 2 weeks of the absence will result in a 10-point deduction from your final grade. Late assignments lose 10% per day, but please note that turning in your work late is much better than not turning it in at all. A 10% deduction is minor in comparison to a 0 grade. More details about all of this information can be found on the syllabus PDF version, which you can find on Canvas. The Phage Genomics Lab Canvas site can be reached by signing on to Canvas and clicking on the MCDB 1161 panel from your dashboard. The home page contains many important links that you will use during the semester. By clicking on the syllabus PDF link, you can find a PDF copy of the syllabus as shown earlier in this video. The modules link links to the assignments for each week and submission links for some assignments. The modules are organized by weeks. Within each week are links to the pre-class reading assignment and videos. Not all weeks have videos. The lab activities for the week, which are found in your lab manual, and hard copies are turned in to your TA, and links for submission of any assignments that are turned in digitally, for example, the Week 1 in-class assignment. Additionally, you will find links to the pre-class assignments and problem sets, which consist of multiple choice questions that are submitted through Canvas. The Learning Objectives links to a list of the course learning objectives. These learning objectives make a great study guide for the midterm and final exams. The Contact Information link links to contact information for all instructors, TAs, and LAs. The Open Lab schedule lists the time of scheduled open labs and the LA scheduled for each open lab time. The Important Date link links to the due dates for the major assignments in this course, including the paper drafts and final paper, and the poster drafts and final poster. The course files, such as PowerPoints, lab data, and practice assessments, can be accessed through this course files link. Finally, the Wednesday lectures are recorded by Lecture Capture and can be viewed by clicking on the lecture videos link and entering your identity credentials. These videos are a great way to catch up if you miss a lecture or want to review a particular topic.
Hello, I'm Nancy Guild, and I'm the other instructor for the Phage Genomics course. Our lab space is relatively small and shared with many students. For everyone to have a positive research experience and for this course to run smoothly, please use the following guidelines at all times. When in doubt, ask. Your LAs, TAs, and instructors are here to help you and answer any questions you have, no matter how insignificant you may think they are. Please keep the lab clean. Wash common equipment every time that you use it. This includes like cleaning the balance or rinsing out gel boxes. And if you spill something or if your auger overflows in the microwave, clean it up immediately. Use your own supplies or designated common supplies. Make sure all your own supplies are clearly labeled with your name, date, lab section, and description of contents. This needs to be done for every microfuge tube, test tube, or bottle that you intend to keep for more than a day. Remember to keep your sample tubes and auger plates in this proper section of the incubator and refrigerator, which will be specifically labeled by lab section number. Be sure to put your microfuge tubes in the sample storage boxes labeled with your section number. Because of the large number of students and samples, please properly dispose of your samples when they are no longer needed. Remember, when you leave the lab, to make sure that all gas supplies to the Bunsen burners are turned off and the Bunsen burners are put away. Make sure that your bench is cleaned up.
Your lab notebook should include the following for each day you complete experiments. The title, purpose, and date are required for each day that you work in the lab, even if you are continuing or repeating an experiment. The purpose is the reason for doing the experiment. Note that some experiments have more than one reason that you might complete them, but in this section you should just highlight the reason you are doing it this particular time. Your notebook for each class should start with the results from the previous class. Include all relevant information about your results, including which plates did not have plaques, the size and morphology of plaques, description of gel results, etc. Don't forget to include results for every plate and experiment, even if you aren't using that plate or experiment for future experiments. A picture or drawing should be included for most results. Draw a line down the middle of the page. On one half, write the procedure, and on the other half, write your lab notes as you are actually performing the procedure. Summarize the procedure from your lab manual. You do not need to copy the protocol word for word from the lab manual. Use a shorthand that makes sense to you, and make sure that it is complete enough that you could do the experiment just looking at your lab notebook. After the first time you do a protocol, you do not need to write it out in your lab notebook. For example, write, "For protocol, see page 27. Lab notes should include any information you notice while you are doing the experiment, how the sample looks, any deviations from the protocol, exact time samples were incubated or centrifuged. This section should never be blank, there are always observations, and this information may be essential for troubleshooting in future experiments. In the future directions, describe what you will do in the next lab period. This section should be a brief summary of your future plans.